Okay, welcome back. We are starting a new chapter. We're jumping back to chapter 12, which is nervous system or nerve tissue. The other control system. Again, like I mentioned, primary communication control system, rapid response. Remember the differences between the endocrine system and the nervous system. Nervous system is generally really rapid. Information control. So it's getting information, analyzing information, making a response. So as you just said, collect information from receptors. So you acquire information from your receptors. You process and evaluate that information at a control center. Oftentimes it's the brain. And you make decisions about what to do about that. Sometimes it's a conscious decision, lots of times it's unconscious or involuntary decision. And then you initiate response to the information after you've evaluated to the effectors to do something about it. Okay, we can talk about divisions of the nervous system. One would be the central nervous system, or CNS. And that's the brain and spinal cord. Then there's the peripheral nervous system. Of course, that's everything outside of the brain and spinal cord. So, and we're going to divide the peripheral nervous system, or PNS, into two parts, sensory and motor. And then we're going to divide sensory up into two parts. So here's the thing. Information from the peripheral nervous system comes into the central nervous system. And that's sensory, right? And then there's visceral sense and somatic sense. Well, I didn't put the microphone on, did I? I actually don't think I need a microphone. Okay. Or that microphone, I should say. Somatic sensory is something you're aware of. Okay. Visceral sensory is something that you're not aware of. It's sensory information, but you're never conscious of it. Then there's motor, and that can be somatic and visceral also. Somatic motor is voluntary movement, right? Visceral motor is well, sort of like involuntary movement. You are not controlling that movement, right? And there are two divisions of this visceral motor, also known as the autonomic nervous system, sympathetic division and parasympathetic division. And they're all sort of equally, they're all partially working at all time, but one will be more dominant over the other depending upon what situation we're talking about. And we'll talk a lot more about that later on. Oh, I should have brought the book in. This is right out of the book. I didn't bring the book in, so I can't tell you what page it's on. So it's a structural organization of the nervous system, as you mentioned. CNS, central nervous system, PNS, peripheral nervous system. And then it's functional organization. Sensory nervous system detects stimuli and transmits information from receptors to the CNS. So remember, sensory is from the periphery into the CNS. Motor is from the CNS to the periphery. Somatic sensory input that is consciously preserved from receptors, what's called the special receptors, which is the ones around your head, and the general receptors that are all over your body. Eyes, skin, ears. Visual sensory input that is not consciously perceived from blood vessels and internal organs, like heart and GI tract. That's visceral sensory. Then the motor nervous system, remember, initiates and transmits information from the CNS to effectors in the periphery. Somatic motor is motor output that is consciously or voluntarily controlled. So the effector is skeletal muscle. Autonomic motor output that is not consciously or is involuntarily controlled. Effectors are cardiac muscle, smooth muscle glands. Okay, universal properties of neurons. All neurons have these properties. Excitability, conductivity, secretion, longevity, and amitotic. Excitability, ability to generate an action potential. Now, we're going to be talking a lot about potentials in this chapter. And a potential is just really a separation of charge, ability to do something. So the first one we'll talk about action potential. So the ability to generate an action potential is called excitability, and there's only two tissues that can do that: nerve tissue and muscle tissue. Conductivity is the ability to conduct this nerve signal, this action potential. Secretion is what happens most of the time when two nerve cells talk to each other, or a nerve cell talks to a muscle cell, or a nerve cell talks to a gland cell, by secreting a signaling molecule. Remember, when we put up the endocrine system, we found out that that's something the endocrine system and the nervous system have in common. They both secrete these molecules. But in the nervous system, the secreting cell is here, the receiving cell is right next to it. And these signaling molecules are called neurotransmitters. Longevity, they last a long time, hopefully your entire life. And you want them to last your entire life because you don't get them back if you lose them, for the most part. A couple of exceptions there, but generally you can't replace nerve cells because of the next property. They are amitotic. Now we're talking about adults, and I'm talking about fetuses, infants, adolescents. They're still making nerve cells. Once they get to adult, you don't make any more nerve cells. As I said, a couple of exceptions. We'll talk about those exceptions when we get into the brain. One area in the brain, it's an exception. And in one area in the senses, that's an exception. But for most part, they don't go through mitosis, no cell division anymore with nerve cells. Parts of a neuron. Almost all neurons, except some really strange ones, have all of these parts. Dendrites are where information is received. Soma is also known as the cell body, is where the nucleus and all the organelles are. The axon hillock is sometimes called the trigger zone. That's where an action potential can be generated. The axon is the nerve, well, not really a nerve. Nerve is a bundle of axon. Is the part of the nerve cell that transmits the action potential. Then an axon terminal is the what's called the postsynaptic part of the cell. It forms a synapse, which is the signaling part, and that's the part of the neuron that does the secretion, secretes a neurotransmitter. And most cells, nerve cells, are covered with a fatty sheath called the myelin sheath. It's generally just on the axon. Okay, axonal transport. I'm going to go into axonal transport very much. This is fast and slow. And that is moving things inside the cell, across a cell, down the axon. And that really, a good example I can think of that is the posterior pituitary. It's nerve tissue, remember? And remember those neurotransmitters, ADH and oxytocin, are made in this cell body of cells in the hypothalamus, and they're transported down to the posterior pituitary. And that's axonal transport. And it's a fast one and it's a slow one. That's all we can do about that. Well, there's a couple of neurons there. Drawings of the neurons over here, and that's an actual neuron. And that looks familiar from the tissues lab, because that's how we identified nerve tissue, by looking at those kind of cells right there. And really, this is the axon. These are the dendrites. That's cell body. So cell body, here's all the dendrites. This is called a multipolar cell, and it's lots of dendrites, one axon. The axon can have an axon collateral, so it becomes two, but leaving the cell body, it's just one axon. And the axon hillock is the very first part of the axon. And it's an integrating region, and that's a decision-making region. And the decision is, do we turn on an action potential or do we not? And these nerve cells act like a digital computer. They're only in two states. Now, in this computer that's doing this, there are decisions being made, and it's all a matter of either something's on, which is a 1, or it's off, which is a 0. And all the information in those computers, it just comes down to a digital on-off. Is this switch on? Is this one off? It's a series of on-off switches. Nerve cells the same way. It's either on, which is generating action potentials when it's on, or it's not. If it's not generating action potential, it's off. And the decision is made at the axon hillock, and the decision comes down to bioelectricity, which we're going to get into later on. Now down here are the axon terminals, or synaptic knobs, and they are synapsing upon dendrites or cell body of another cell, another nerve cell. And there's a blow up of what's called a chemical synapse right there. Okay, anatomic types, that should be of neurons, not or neurons. Anatomic types of neurons. There's lots of different types of shapes of neurons and everything, but they kind of fall into 4 categories. And it really comes down to how many processes. So processes are things coming out of or to the lengths of protoplasm leaving or coming to the cell body. Multipolar means lots of processes on the cell body. Bipolar means 2 processes on the cell body. Unipolar means 1 process. And inaxonic is a really bizarre type. I think there's only one example, and that's no axon. So they're missing part of the, actually no axon terminals either. So, well, we're gonna show you a picture, I think I showed you pictures in later slides of these 4 types. No, of three of types, not the 4th one. Functional classification. So, a sensory neuron is bringing information from the periphery into the central nervous system. So its dendrites are out in the periphery, gathering information and sending information along an axon into the central nervous system. This is also called afferent. And part of the axon, now the cell body and the axon and the dendrites are in the PNS. The last part of the axon and the axon terminals is in the CNS. And this is called afferent. Motor neurons are the opposite. They're efferent, bringing information from the CNS to the PNS. So their dendrites and cell body are in the CNS, and the first part of their axon is in the CNS, but the rest of the axon and the axon terminals are in the PNS. Then there are neurons whose in the entire cell exists within the central nervous system, and that's called an interneuron. And it's a classic reflex, which I'm going to show you, about information coming from the periphery, going into the central nervous system, a decision being made, information coming out of the central nervous system to an effector causing a change. And a lot of these reflexes, and we have a chapter later on there's going to be a lot more about these reflexes, that they're often unconscious. You don't really think about it. And they're often protective. So here it is right here. Here's skin receptors. So these are dendrites out there. They're pressure receptors, really. Let's say someone comes around and taps you on the shoulder over here. That information, it's sensory input, goes into the nervous system through what's called a posterior root. And this is a posterior root ganglion. Now, ganglion is a collection of cell bodies outside the central nervous system. And that's a cell body of the sensory neuron, and then sends its axon in, and it synapses upon this purple guy, which is an interneuron. The interneuron is stimulated to fire an action potential, and it has synapses on a motor neuron here. And there's the dendrites of the motor neuron, cell body of the motor neuron in the central nervous system. Here's the axon leading up, and this interneuron stimulates that motor neuron to fire. It sends out information along its axon to skeletal muscles, which makes you turn around. And actually you do that automatically. You would react. Something came along, touching your shoulder, you would react. And actually, after you react, you find out that you did react. Oh, somebody or something touched me on the shoulder. So one thing that's not shown here is that there's an axon collateral, probably from the interneuron, that's going out into this area here. This is called the white matter. This is the gray matter, and here this is called white matter. And these are axons that are ascending and descending the spinal cord. So there's going to be information sent out going to your cerebral cortex and informing you that something just touched you on the shoulder. So this is, and it could be protective because it could be something dangerous touching you on the shoulder. You're going to be reacting and get ready for it. Okay, nerves, bundle of axons. And remember, a ganglion, and these nerves are peripheral. Nerves are in the PNS. If there's a bundle of axons in the CNS, we call it a tract instead. So, and then bundle of cell bodies in the PNS are called ganglion. They're called a nucleus in the central nervous system. There are coverings over these axons called the epi, peri, and endoneurium. Endoneurium is around the individual nerves. Then there's a peri neurium around a bundle of these axons. And then the nerve itself is a bundle of those bundles, and that's covered by something called the perineurium, which is probably dense irregular connective tissue. Okay, classification of nerves. Cranial versus spinal. Cranial nerve is coming out of the central nervous system. No, wait, that's wrong. It's coming out of the brain. Cranial comes out of the brain. Well, either coming out or going in. Spinal is coming in and out of the spinal cord. Right? And what's coming in or going out is whether it's sensory or motor or mixed. So if it's a nerve that's just gathering information coming into your central nervous system, you can think of that as a pure sensory. And like optic nerves would be a good example of a pure sensory nerve. It's just information coming in. Motor would be the exact opposite. It's purely axons carrying information from the central nervous system out to the peripheral nervous system. And there are very few nerves that are pure motor. Because we're going to find out that something called proprioception goes the other way along the same nerves. But, well, the muscle that control your eye muscles, going back to eyes again. So you have the optic nerves bring sensory information into your brain from the periphery, and the little muscles that control your your eyes, where your eyes are, those are voluntary, and those are probably pure motor that just move your eyes. Mixed nerve, now that implies that most nerves are mixed, and that's true, that they're both sensory and motor in the same nerve. And remember, these nerves are insulated from each other, so there's very little crosstalk between them. Two axons going in different directions right next to each other are not going to interfere. Their electrical signals aren't going to get confused. Okay, synapses are how neurons talk to each other. And there's two types, electrical synapses and chemical synapses. Most of the synaptic synapses we'll be talking about will be chemical. There's only a few examples of electrical, and they only have a couple advantages over chemical. Chemical has a lot more advantages, which we'll get to later on. And example of electrical synapse, we've already talked about in the tissue chapter. A gap junction is essentially an electrical synapses. Ions flow directly from one cell into the other and change the what's called the membrane potential of the cell that's receiving the ions. Chemical are synapses where the cells are really close together, and the presynaptic cell secretes neurotransmitters that travel to the postsynaptic cell and affect its electrical activity. So that's a chemical synapse. And we're going to look at the structure of a chemical synapse later on and go into much more detail about how synapses work. Neuroglia cells, or sometimes just called glial cells. These are helper cells in the, they're mostly in the central nervous system. There's several types in the central nervous system. You're going to have to know what they are and basically what they do. And then there are some glial cells in the peripheral nervous system. And like I said, they're helper cells. They help what's called the functional cells. And of course, the glial cells have functions. When we talk about functional cells of the nervous system, we're talking about the neurons, because the glial cells are not excitable. They don't carry X potentials, they do other things. Okay. And these are the types: oligodendrites, astrocytes, ependymal cells, microglial cells are all in the central nervous system. And then neurolemocytes, or also called Schwann cells, are both in the central nervous system, are in the peripheral nervous system. And satellite cells are and those are helper cells in ganglia that are in the peripheral nervous system also. Okay, the myelin sheath. These are actually those neurolemocytes or Schwann cells. They're wrapped around axons and they're filled with this fatty substance called myelin. and they're and we're going to see most of the nerves are like that. There are some fibers or nerves that are unmyelinated, and then the gaps between the two Schwann cells is called the neurofibril node, or it used to be called nodes of Ranvier. I learned them as nodes of Ranvier. And the initial segments are really the axon hillock is the initial segment, also called the trigger zones. It's got several names. Okay, so here are some glial cells. Okay. For one thing, astrocytes, this green one, they coat capillaries in the brain, and they are really the major basis of the blood-brain barrier. They make things really, they're really selective. The brain is a special, like, gated community. It doesn't let anybody in or out. And so remember the capillaries in your, we'll talk about how tight capillaries are in next semester in the vessels chapter. But suffice it to say that your brain capillaries are the tightest because of the astrocytes. Okay. Oligodendrites, they make the myelin sheaths in the central nervous system. And so they're the equivalent of a Schwann cell, a neurolemocyte of the central nervous system. But they make myelin sheaths in a different way than the Schwann cells does, unfortunately. And I'll explain what that unfortunate nature is later on. It gets down to regeneration. Then you have microglial cells, are the COPs. They're the immune cells of the brain. So regular cells of the immune system can't get into the brain, so they have their own police force, and that's called the microglial cells. Finally, the ependymal cells line the ventricles in the brain. And the ventricles are fluid-filled cavities in your brain. And they coat they're inside the brain, and they coat the whole outside of the central nervous system with cerebrospinal fluid, which is very similar to blood plasma, but it's a little different, but it's similar to blood plasma. And it is made by these ependymal cells. Right? Okay, over here in the PNS glial, so the CNS glial cells, PNS glial cells, satellite cells surround the cell bodies of the sensory neurons. And here are the neurolemocytes here, and here are also neurolemocytes or Schwann cells covering the axons, and look at the neurofibril nodes between them. And so this neurolemocyte, the entire cell forms the myelin sheath. It wraps around the axon. Okay. Okay, so one of the major functions of myelin is conduction speed. It really speeds up the conduction of the nerve signal, the action potential. It's conducted in much faster than unmyelinated. And the reason for that is something we'll be talking about in a later slide. Regeneration is a big thing. In the PNS, depends on the integrity of the sheath. In the CNS, very little. And we're going to look at, in a later slide, why if you slice open a nerve in the PNS, you can regenerate that function if it's a nice clean cut. But in the CNS, central nervous system neurons do not regenerate. When you cut an axon in the central nervous system, it does not regenerate, unfortunately. And that has to do with the nature of the myelin sheath. How the myelin sheath is made in the central nervous system Now it's made in the peripheral nervous system. And there it is right there. Okay, in the peripheral nervous system, we have these neurolemmocytes or Schwann cells all along here, right? And each cell is committed to forming each part of the sheath, and the nodes of Ranvier are the spaces between them. Okay. In the CNS, these oligodendrites make the myelin sheaths, and all they do, and their cell bodies are not wrapped around the axons. They send out processes, and the process makes the myelin sheath. Okay. So here's the problem. So let's say you cut this this axon right there. Now the cell body is okay. As long as the cell body is okay, the cell is still alive. Okay, but the axon's cut. And if it's a nice clean cut, the axon and axon terminals downstream from the cut will degenerate, and they'll be gone. But the cut end of the axon is still living material and can start regenerating, sending out tendrils from the cut end, trying to find the myelin sheath. If the myelin sheath is still intact, because you make a nice clean cut, those tendrils can find the opening in a Schwann cell and grow into it. And then they find the next Schwann cell, and the next one, and the next one. And they will eventually find a way to the last Schwann cell and find a way to the target organ, the effector that's going to reform axon terminals and regenerate function. But, well, as long as it's not lacerated, if you get a lacerated destruction of some axons and the myelin sheath is destroyed, that's not going to regenerate. So you got to have an intact myelin sheath. Unfortunately, here in the central nervous system, let's say you cut this axon right here. Whoops, let me go back. This axon right here. Well, then let's say the cell body's over to the left here. Then everything downstream of it will degenerate. And what will happen is the oligodendrites will withdraw these processes. They'll withdraw them back into the cell body. There won't be any sheath left because the myelin sheath is withdrawn. Once the nerve inside that's surrounded by the sheath degenerates, then the sheath is withdrawn. And you can have little tendrils coming out of the cut end of the axon, but there's no sheath to be found. They don't know where to go. And they get lost and they don't regenerate. So that's why spinal cord injuries are permanent, because central nervous system axons do not regenerate. Okay, I'm going to cut it there because it's been about 35 minutes. And this is a whole new topic in nervous tissue, pumps and channels. So I'm going to stop it there and pick this up on the next one. So this will be 203 nerve 1. Okay. Catch you in the next video. Okay, let's stop the recording.